Hello. Today we'll be looking at the Watson and Crick model of DNA they published in 1953. What does the Watson and Crick model look like? Here, DNA is a double helix structure that consists of stacked nucleotides one on top of the other. Key characteristics that are associated with this DNA model are base pairings and base stackings, nucleotide structures, which I have circled here, antiparallel strands, right-handedness, and major and minor grooves. All of these will be looked at further in the presentation. First, we have nitrogenous base pairs consisting of thymine, adenine, cytosine, and guanine. Thymine is always paired with adenine, and cytosine is always paired with guanine. This is called a complementary base pairing. They are partially linked together via hydrogen bonds indicated by the dots. Cytosine and guanine have 3 H bonds, while thymine and adenine only have 2. Thymine and cytosine can be described as pyrimidines, while adenine and guanine are called purines. They are separated into these groups because thymine and guanine are similar in structure since they only have 1 ring structure, and guanine and adenine have 2 ring structures. Per the middle photo, the bases are paired together with their complementary partner, then stacked on one on top of the other. This helpful towards the structure of DNA since the nitrogenous base are hydrophobic, meaning they don't like water, and they are huddled closer together, maintaining the internal structure of DNA. The structure of a nucleotide consists of a phosphate group, a deoxyribose sugar, and a nitrogenous base pair group. Nucleotides are what make up the physical structure of DNA. To help keep the structure intact, the phosphate backbone keeps the DNA shape, consisting of a phosphate group and a deoxyribose sugar. Each of the carbons on the deoxyribose sugar also play an important role in DNA. The first prime carbon attaches onto the nitrogenous base. Second prime determines whether the nucleotide is for DNA, which is determined by a single hydrogen, or RNA, which is determined by a hydroxyl group. The third prime is responsible for attaching onto the next nucleotide of the 5' end, and the 5' carbon attaches to the phosphate group. Here we have 2 other characteristics, antiparallel strands and handedness. Antiparallel strands is the idea that phosphate backbones on each side are facing the opposite direction. So on the left side, we have 5' to 3', and on the right side, we have 3' to 5'. Every DNA structure should be on the right-handedness, as you see here on the DNA structure to the right. Handedness refers to the way that DNA is rotated or spiraled around. As you can see on the right-handed one, it goes up and to the right. This is the correct spiral for this DNA model. The grooves along the DNA are gaps along the double helix model. The larger ones are considered the major grooves, while the smaller ones are considered the minor grooves. This is caused by the orientation of nitrogenous bases along the phosphate backbones. Lastly, how would salt ions affect the melting temperature and structure of DNA? Well, since the phosphate groups are negatively charged, the addition of salt ions would then neutralize them, increasing the integrity of the structure. In turn, making it harder to denature the DNA with limited heat, which then increases the melting temperature of DNA. These are the resources that I use for this.